Scene script. Have you ever wondered how the tiny chips in your smartphones or medical devices work? The answer lies in microfluidics. Microfluidics, in its essence, is the science and technology of systems that process or manipulate minute amounts of fluids, typically in the range of microliters to picoliters, using channels with dimensions in the micrometer scale. It's a field that's as fascinating as it is intricate. This field has revolutionized the world of technology, bringing forth a new era of precision and control. It's like having the power to manage an entire chemical laboratory on a chip, no bigger than your thumb. Imagine being able to regulate the flow of fluids, akin to traffic control at a microscopic level. That's the magic of microfluidics. But why is it so significant in today's technology? Well, microfluidic systems offer a high degree of control over physical and chemical processes. They allow for rapid, highly parallel and portable operations, which are extremely desirable in many applications. From healthcare, where they are used in lab-on-chip devices for disease diagnostics and drug discovery, to the food industry, where they help in precise flavoring and packaging, microfluidics is everywhere. Furthermore, microfluidics has also found a place in environmental monitoring for detecting pollutants and microorganisms, and in the field of tissue engineering and organ-on-chip technologies. It has even ventured into the realms of nanotechnology, where it has been used for the synthesis of nanomaterials. One of the most intriguing aspects of microfluidics is its ability to deal with single cells, making it a potent tool in the world of biological research. It has paved the way for groundbreaking discoveries in cell biology and genetics, leading to advancements in personalized medicine. So when we talk about microfluidics, we're not just talking about a scientific field, we're talking about a transformative technology that has the potential to change the way we approach health, food, environment, and even our understanding of life itself. So microfluidics is the backbone of many modern technologies, providing precise control over fluids at a microscopic level. The magic of microfluidics happens inside tiny devices, with intricate components. These devices are micro-engineered platforms that guide small volumes of fluids through a network of microchannels, pumps and valves. Now you may think of channels as the highways of microfluidics, guiding fluids in a controlled manner from one point to another. Then we have pumps, the heart of the system, driving fluid flow through these channels. They can be mechanical, like peristaltic pumps, or non-mechanical like electrokinetic pumps. Each has its unique advantages and applications. Next, the valves, the traffic controllers of our microfluidic highway. They regulate the flow, directing it where it's needed when it's needed. These can be passive, relying on fluid properties and channel geometry, or active, controlled by external forces. These tiny yet sophisticated components work together to manipulate fluids in incredibly precise ways. They make microfluidics a versatile tool for a plethora of applications, from medical diagnostics to environmental monitoring. Now, how does fluid behave at such a small scale? Welcome to the fascinating realm of microscale fluid mechanics, where the fluid behavior deviates significantly from what we observe in our day-to-day life. At the microscale level, the world of fluids is governed by a different set of rules. Let's start by addressing the elephant in the room, the Reynolds number. This dimensionless quantity is a significant player in the dynamics of fluid flow. At the microscale, the Reynolds number tends to be quite low, indicating that viscous forces dominate over inertial forces. This results in a phenomenon known as laminar flow, where the fluid flows in parallel layers with no disruption between them. Next in line is the concept of surface tension, which becomes a dominant force at the microscale. The intermolecular forces within the fluid lead to this surface tension, making the fluid behave more like a stretched elastic sheet rather than a liquid. This can lead to interesting effects such as capillary action, where fluid can flow against gravity in narrow spaces. Let's not forget the importance of diffusion in microscale fluid mechanics. At these small scales, diffusion becomes an efficient means of mass transport. This is in stark contrast to the macro scale, where convection is typically the primary mode of transport. Finally, the concept of slip at the fluid-solid interface is worth mentioning. In macroscale fluid mechanics, we often assume no slip conditions, that the fluid at the boundary is stationary. However, at the microscale, this assumption may not hold true, leading to slip conditions where the fluid velocity at the boundary is not zero. These principles of microscale fluid mechanics, low Reynolds number, dominance of surface tension, efficient diffusion, and potential for slip conditions, drastically alter the fluid behavior. They require a shift in our understanding and approach towards designing and working with microfluidic systems. Understanding these principles is key to designing effective microfluidic devices. Microdroplets and microcapsules play a significant role in microfluidics. But what are they exactly? Let's dive into the fascinating world of these minuscule entities. Microdroplets are tiny droplets, typically in the range of micrometers to millimeters in diameter. They are formed when a fluid is dispersed into another immiscible fluid. Imagine a droplet of oil in water, or a droplet of water in oil. The process of forming these droplets is called emulsification, and it's where the magic happens in microfluidics. Now, what about microcapsules? They are similar to microdroplets, but with an added layer of complexity. A microcapsule is essentially a microdroplet that is encapsulated within a thin, solid shell. This shell can be made of various materials depending on the application, from polymers to proteins, and even metals. The encapsulation process provides a protective barrier for the contents of the microcapsule, allowing for controlled release or isolation of their contents. So, where do these microdroplets and microcapsules fit in the world of microfluidics? Well, they are the key players. Microfluidic devices often rely on the formation and manipulation of these microdroplets and microcapsules. They can act as individual reaction vessels, where chemical or biological reactions can take place in isolation from each other. This can drastically increase the throughput of experiments or reactions, while reducing the volume of reagents used. Moreover, the precise control over the size and composition of these microdroplets and microcapsules in microfluidic devices has opened up new opportunities in various fields. From targeted drug delivery, where microcapsules can deliver drugs directly to a specific location in the body, to single-cell analysis, where individual cells can be encapsulated in microdroplets for study, the applications are vast and varied. In essence, these tiny droplets and capsules are the workhorses in a microfluidic system, carrying out specific tasks with precision. They make microfluidics a versatile tool, pushing the boundaries of what we can achieve in fields like medicine, chemistry, and biology. These tiny droplets and capsules are the workhorses in a microfluidic system, carrying out specific tasks with precision. Microfluidics isn't just about physics. It's also making waves in biology and medicine. Picture this: tiny devices manipulating minute amounts of fluids, and these tiny droplets or streams are used to perform biological or chemical processes. That's what biomicrofluidics is all about. Biomicrofluidics is a burgeoning field that brings together biology, medicine, and microfluidics. It's a field that's been gaining traction because it has the potential to revolutionize how we diagnose diseases, deliver drugs, and even engineer tissues. Think about disease diagnosis. Traditional methods can be invasive, time-consuming, and require a substantial amount of samples. Biomicrofluidics, on the other hand, offers a less invasive, faster, and more efficient approach. It allows us to analyze small volumes of biological fluids like blood or urine to detect diseases at their earliest stages. Now, let's talk about drug delivery. Getting drugs to the right place at the right time in the right amount is a delicate balancing act. Biomicrofluidics can help tip the scales in our favor. It provides precise control over drug delivery, making treatments more effective and reducing side effects. And then there's tissue engineering. Imagine being able to grow tissues and organs in the lab. Sounds like science fiction, right? But with biomicrofluidics, it's becoming a reality. By controlling the microenvironment, we can guide the growth of cells and tissues. This could lead to breakthroughs in regenerative medicine and organ transplantation, a true game changer in healthcare. So, you see, biomicrofluidics is more than just a scientific curiosity. It's a field that's pushing the boundaries of what's possible in biology and medicine, and we're only scratching the surface. As we continue to explore and innovate, who knows what other breakthroughs biomicrofluidics will bring? Biomicrofluidics is revolutionizing healthcare, opening up new possibilities for patient treatment and research. So let's keep our eyes on this fascinating field as it continues to evolve and shape the future of medicine. Despite its potential, microfluidics also faces some challenges. As we delve into the intricate world of microfluidics, it's important to acknowledge the obstacles that exist within this field. One of the major challenges has been the difficulty in fabricating microfluidic systems. The creation of these tiny, intricate devices requires precision and meticulousness, which can be time-consuming and costly. The nature of the materials used, such as polymers and glasses, also presents its own set of challenges, as they can be sensitive to temperature and pressure changes. Then there's the issue of scaling. While microfluidic systems offer a high degree of control at a microscale, scaling these technologies to an industrial level is not straightforward. The problem lies in maintaining the precision and control that make microfluidics so attractive when operating at larger scales. Despite these challenges, the field of microfluidics continues to evolve and expand. Researchers are developing new materials and fabrication techniques to overcome these hurdles. For instance, the use of 3D printing technology is being explored to create intricate microfluidic devices more efficiently. Looking ahead, there's a growing interest in integrating microfluidics with other technologies, such as nanotechnology and bioengineering. This interdisciplinary approach could open up new avenues for innovation, allowing for the development of more sophisticated systems capable of performing complex tasks. Moreover, the potential applications of microfluidics are vast and varied. From drug delivery to environmental monitoring to food and beverage production, the possibilities are seemingly endless. It's this potential that continues to drive research and development in the field, despite the challenges. Lastly, there's a push towards greater automation and control within microfluidic systems. This could lead to the development of smart microfluidic devices, capable of self-regulation and adaptation, further expanding the potential applications of this technology. While challenges exist, the future of microfluidics is bright, with endless possibilities for innovation. Despite the hurdles, the field continues to evolve, driven by a desire to push the boundaries of what's possible and to harness the potential that microfluidics holds. So, what have we learned about microfluidics? Our journey through the world of microfluidics has taken us from the basic principles to the cutting-edge applications. We have delved into the intricacies of microfluidic devices and components, understanding their key role in manipulating fluids at the microscale. Fluid mechanics, a vast field in itself, takes on new dimensions when applied to the tiny world of microdroplets and microcapsules. Our exploration of microdroplets and microcapsules has revealed their potential in a myriad of applications. From drug delivery systems to environmental monitoring, the use of these tiny droplets and capsules is revolutionizing industries. We also dove into the realm of biomicrofluidics, where biology and microfluidics intersect. Here we discovered the potential for microfluidics in point-of-care diagnostics and single-cell analysis, revolutionizing healthcare and medical research. The potential of microfluidics extends even further. We touched upon organ-on-a-chip technologies, where microfluidic devices replicate human physiological responses, providing new avenues for drug testing and disease modeling. The food and beverage industry, environmental monitoring, bioprinting and tissue engineering, nanomaterial synthesis. All these fields are being transformed by the power of microfluidics. But it's not just about what microfluidics can do now. We also looked ahead to the future, discussing the challenges and opportunities that lie on the horizon. Microfluidic automation and control, for instance, holds great promise for further advancements in this field. It's clear that microfluidics is more than just a scientific curiosity. It's a tool, a medium, a technology that is shaping our world in ways we could barely have imagined just a few decades ago. And as we continue to innovate, to push the boundaries of what is possible, microfluidics will undoubtedly be at the forefront of many of these advancements. As we continue to push the boundaries of technology, microfluidics will undoubtedly play a crucial role in shaping our future.